everybody. Good morning, good afternoon, good evening, depending on where you're joining us from. I'm Diana Mitra, Head of Scientific and Corporate Communications here at DuPont Therapeutics, and welcome to Kitchen Table Talk number 39. And we welcome you here together with my co-host, Cameron Rizzolo, an Associate Principal Scientist in the Chemistry and Drug Discovery Department here at DuPont. And we're here in the kitchen table in Boston. So before we begin, I have a few housekeeping announcements. So this is a live event, so please be courteous. Keep your microphone muted. Keep your camera on if you'd like, and not along with the speaker. And enjoy the talk. During Q&A, we're going to call you and ask you to unmute. At that point, please unmute your microphone and ask your question. Ideally, we would keep the questions to the end, or at least to the breaking points that John will have during his talk. So after every section, there's going to be a break for Q&A. So without further ado, I'm going to pass the baton to Cameron to introduce today's speaker.
Thanks very much, Diana. So it is with great pleasure that I get to introduce today's KTT speaker, Dr. John Ditlev. So I first met John during my postdoctoral studies back in 2019 at the University of Toronto. I met him actually at the official condensate journal club, which back in the day was a room with, say, 10 to 15 people, and we would meet like once a month. So now, like 6 years later, my old department, I realized that has a whole class called biological condensates, in which John teaches at, and also other folks that are in the journal club. So it's a reflection of not just how much the field has progressed, but also how it's evolved in my old department too, which is very cool. As we'll hear today, John has done extensive work studying phase separation in neuronal and immunological signal transduction systems. He's a biochemist and biophysicist by training with a PhD from the University of Connecticut. And afterwards, he did his postdoctoral studies at the UT Southwestern Medical Center with Professor Michael Rosen. His work there centered on phase separation and the stoichiometry within as a way to explain signaling activity in actin filament clusters and signaling during T cell activation. John's current role is as an assistant professor at the Department of Biochemistry at the University of Toronto, and he also holds a position as a scientist at the Hospital for Sick Children. There, he continues to do groundbreaking work to study the association between phase separation, that is at the level of biochemical reconstitution, but also cell biology, in membrane receptors and signal transduction, both in neurons and immune cells. In addition, his team now looks at the effect of phase separation on local RNA translation and interactions between different condensates. So I'm excited to hear more about the evolution of this story. So without further ado, please join me in welcoming Dr. John Ditlev. The floor is yours, John.
All right. Cameron, thank you for the very kind introduction, and Deanna for organizing this talk, and also for everyone that's here for taking time out of your day to hear some of the work that's going on in my group. Okay, and so today the goal that I have is to talk about some of this work that we've done looking at this postsynaptic density protein shank2 and how missense mutations in its intrinsically disordered region can affect its some of the biophysical properties and the function of the condensates that form. But before I begin, I'd just like to acknowledge the people who actually do the work in my group, specifically Dr. Lianagi, who spearheaded this project shortly after joining my group as a postdoctoral fellow. Steve, Gaddy, Shusheng, Victoria, Anna, and Sam have all done work as well, assisting Lashani in her project. And then, of course, thank the funding agencies that fund our work. Okay, so just for people that maybe are joining us that are not as well-versed in condensates as much of the audience, I thought I would just talk a little bit about cellular organization. And so historically, cellular organization has been thought of as being the cell is organized by membrane-bound compartments, such as mitochondria, the endoplasmic reticulum, the Golgi. But more recently, in about the past 15 years, there's been a lot of work that's shown that there are many non-membrane bound compartments that also form, and these are termed biomolecular condensates. And as I want you to see, they can form in the nucleus, in the cytoplasm, but also on membranes. So even though they're non-membrane bound, they can be membrane associated. And just as some examples of well-described condensates include the nucleoli, PML bodies, P bodies, as well as T cell receptor clusters, which I'm going to use as an introduction to membrane-bound condensates. And so how do these biomolecular condensates form? Well, they usually form through multivalent weak interactions, although some interactions can be very strong as long as they're highly dynamic. And so as an example, I've got the blue molecule, multivalent blue molecule, and multivalent orange molecule here. And these can undergo two types of interactions. The first is a high affinity interaction in which the two biomolecules will zipper, and the result of that is the formation of a heterodimer. However, they can also undergo these multivalent interactions and form a protein or protein nucleic acid interaction network. When interactions between the proteins or nucleic acids become more favorable than the solvent, these networks can undergo phase separation and the formation of a liquid within a liquid. Importantly, not all phase separation forms liquid condensates, as you'll see later in my talk. There's condensates that form liquid rapidly, but then rapidly undergo a transition into a gel-like state. And this has been shown now for roughly about 15 years. Let's see here. Okay. And importantly, as I mentioned earlier, phase separation can occur both in solution and on membranes. And so if we take this multivalent protein that's phosphorylated in 3 spots, bound by an adapter protein, which is bound by another adapter protein, we can form condensates on membranes if one of the proteins is attached to the membrane. If we take those same proteins and mix them in solution at high enough concentrations, we can form condensates in solution. One of the important aspects of this is that condensates on membranes form at about an order of magnitude lower concentration than in the cytoplasm, than in solution or in the cytoplasm. And this allows cells to organize condensates on membranes at much lower concentrations than are usually required or usually we think of in solution. And so as an introduction to membrane-associated condensates, I want to tell a story that Dr. Xiaolei Su and I spearheaded when we were postdocs. And this is how phase separation regulates biological processes at the T cell immunological synapse. And so at the synapse, we can see here between a junction between 2 cells, the antigen-presenting cell in blue, the activated T cell here shown in red, and then where these 2 cells contact one another, this protein ZAP70 localizes from the cytoplasm of the T cell to the membrane. And at the membrane, ZAP70 phosphorylates this adapter protein LAT, as you'll see in a second, and that is thought to drive signaling at the immunological synapse. And so if we take an on-phos view using turf microscopy of a T cell and look at what happens when a T cell receptor contacts an MHC peptide, or in this case, an antibody, we see that the T cell receptor, that binding activates kinases at the membrane. Those kinases phosphorylate proteins, including LAT. LAT microclusters form. And if you watch the video, you'll see that LAT microclusters form at the edge of the synapse and then are moved towards the center of the synapse. And that this process is actually very important for T cell function and the proper response of the T cell to antigen-presenting cells. These LAT microclusters have been linked with calcium influx, actin polymerization, and also downstream ERK phosphorylation through genetic studies. And so Xiaolei and I asked some questions. How do these clusters form? Is phase separation possibly the mechanism that drives their formation? And also, is phase separation responsible for some of these functions that have been previously described in the literature? And so genetic work has shown that there are many, many proteins involved in T cell activation. I'm just highlighting a few of the proteins here in the signaling pathway, particularly kinases that phosphorylate the T cell receptor and LAT, adapter proteins that bind LAT, as well as more adapter proteins that can activate actin polymerization. And so the first question that Shaoli and I asked is, what are the minimum requirements for phase separation of LAT, if it is in fact phase separation? And so what we did is we attached a fluorescently labeled LAT to a model lipid bilayer, and then we dropped Grab2 and sauce into the solution. And when we did that, we saw the rapid formation of clusters. And further experimentation showed that these clusters are fully consistent with phase-separated condensates. And so we then asked, okay, so we can do this in vitro, but is this important in cell biology? Because there's a lot of biochemistry that we can do that has very little relevance towards what happens in a cell. And so what we did is we took LAT knockout T cells, we re-expressed LAT with different numbers of tyrosines on it, and then looked at whether these different LAT species could form clusters on the cell membrane, as we saw in vitro. And what we found is a very high correlation between our cellular results and our in vitro results. And furthermore, we linked that condensate formation with ERK phosphorylation, showing that condensate formation is correlated with successful cellular signaling in T cells. And this will be very important for what I discuss later in the talk. We also found that LAT clusters could sort molecules. And so in these experiments that were done on model bilayers, we formed LAT clusters and had the phosphatase CD45, which targets LAT in T cells, also attached to the membrane, which is where it's localized in cells. And what we found is that LAT clusters could exclude CD45 from their internal environment, thereby protecting LAT from dephosphorylation, allowing the clusters to be maintained on the membrane. And so we did some further work and found that this was an electrostatic repulsion. That CD45 is a highly negatively charged protein, and LAT clusters are highly negatively charged protein clusters, and the electrostatic repulsion actually allowed for the sorting of these molecules. We also linked LAT cluster formation directly with actin polymerization. And so in this movie, you'll see upon addition of ATP, ZAP70 comes to the membrane, LAT clusters form, and then actin is polymerized from the clusters. And Lindsay Case did some very nice work in a paper back in 2019 showing that it was actually the dwell time of these actin nucleation promoting factors, as well as ARP23, that allowed for the formation of these actin filaments at the LAT clusters, and that this was regulated through phase separation. Okay, so another thing that came out of this extended study that we continued looking at was we asked how these LAT clusters are moved by the cell. More generally, how these LAT clusters interact with their local dynamic environments. And one of the first things that we noticed is that LAT cluster composition changes as it moves from the edge of the synapse towards the center of the synapse. And you can see this here. LAT is present in the clusters as it should be from its inception to where it enters the center of the immune synapse. However, the adapter protein NCK seems to dissipate as the LAT cluster gets closer to the center of the synapse. So we asked what function might that have? And so we took a look at dynamic actin networks, and John Hammer did some really nice work with this in his group. And what you'll see here is that at the T cell synapse, an on-phos view of the T cell synapse, there's two dynamic actin networks. The first network is an ARP23 generated network in which dendritically branched actin filaments form at the membrane and then push backwards. There's also a foramen-generated actin cable network at the center of the synapse that telescopes inward, leading to directed motion across the synapse. So we asked whether this change in LAT cluster composition might allow the LAT clusters to be dragged from the edge of the synapse and then pushed towards the center. And so if you recall, right at about this edge where the two networks transition, NCK falls off. So NCK and NWASP are not included in the clusters at this point. We reconstituted this using dynamic actin networks on model membranes with members from G2 Mayor's group and Ron Vale's group. And Khalid Jacquemon did a really nice job analyzing this data for us. And what we found is that complete clusters, or clusters that have NCK and NWASP, bind to and wet actin filaments, while minimal clusters, as would be found near the center of the synapse, do not tend to bind and wet filaments to the same degree. When we induce motion through myosin activity, we find that complete clusters with NCK and NWASP are dragged with the filaments towards the center of these actin asters, while the minimal clusters containing only GRAB2 and SOS are pushed around by the actin filaments. So this to us generally indicates that condensate composition can not only determine the function as we all think about it, but it also can determine the interaction with local environments, which likely is important for the neuronal synapses, as I'll talk about in a minute. So I'd like to take a break and ask if anybody has any questions about some of this introductory work that Xiaolei and I did previously. I see Jeff has a question.
Yeah, first of all, it's really good to see you, and it's nice to see this kind of replay of this early work, and I have a couple of students with me, so they're enjoying it, I'm sure. The question I have is, have you ever looked at extracellular signals? Like you had LCK in your first cartoon, and LCK is, associated with CD4, and obviously we know CD4 is important for T cell recognition signals. Do those played a role in what's happening inside the membrane of the cell?
Yeah, so this is actually, I think, an underappreciated aspect of T cell signaling. John Hamburn and Jan Burkhardt have done some nice work looking at how the tension of the opposed membrane and opposed cytoskeleton, how that affects T cell signaling. But by and large, there has not been much work looking at how the organization of the opposed cell that might activate CD4 or bind CD8 or PD1 or CD28, how that organization plays into T cell signaling. And I think this will be important not only for understanding T cell signaling, but also for CAR T cell work that my colleague Xiaolei has really spearheaded in his group. So that's a very, at least in my mind, an active question, is how does the opposed membrane regulate what's happening in the T cell, and carrying that over to neurons as well in the future.
Cool, thanks.
Yep. All right.
From the
Any
kitchen
questions?
table.
Okay, perfect. So you came to a neuronal talk, and all I've talked about is T cells to this point. So why have I started this introduction? Well, back in 1968, Gerald Edelman suggested that there may be similarities between immunological synapses and neuronal synapses, and that we probably can learn something about neuronal synapses from immunological studies, as well as learn something about immunological synapses from neuronal studies. And so, this was long before biological phase separation came to the forefront of our minds. And so, me and a few other people have asked the question, well, might phase separation be a general mechanism that regulates the functional organization of both immunological and neurological synapses? So I just showed you that phase separation plays a very key role in immunological synapse formation and function. Can we apply what we've learned in the immunological synapse to neurological synapses? And so what is the neurological synapse that I'm going to be talking about? And so I'm specifically focused at the connection between an incoming axon and dendritic spines on a receiving neuron. So neurons in the brain have roughly 10,000 dendritic spines per neuron, which means that they are able to integrate a lot of information coming in and generate an action potential, which they shoot out an axon. Within the neurological synapse, there's two terminals: the presynaptic terminal, which contains several condensates, including the active zone, synaptic vesicle clusters, as well as some other RNA granule type structures. And then there's the postsynaptic terminal, which is also filled with condensates, including the postsynaptic density, RNA transport granules. There's GIT-PIX condensates that Ming-J Zhang has shown. And if you want more information on this, Kalu Jackman and I wrote just a brief description of how condensates play a role in signaling in the dendritic spine, but also Ming-J Zhang's group has done some really nice work and written a couple of really nice reviews about how condensates can control the passage of information from one neuron to the receiving neuron. Now, I just want to highlight the postsynaptic density. So there's been quite a lot of genetic work done on this structure. And roughly, proteomic work has shown that there's at least 1,000 proteins that can localize to this membraneless structure. And perhaps most strikingly, over 60% of the proteins that localize to the postsynaptic density have been linked with neurodevelopmental and neuropsychiatric disorders. So this might be the most disease-rich structure in the human body, which means that there's a lot of space to operate. And so a closer look shows that the postsynaptic density is scaffolded by multivalent proteins. So there's roughly 17 or so scaffolds that have been recognized to be required for postsynaptic density formation. And specifically for family members. So we've got the Homer family, the McGuck family, which includes PSD95, DL-gap family, which has several family members, as well as the Shank family, Shanks 1, 2, and 3. And each of these proteins can be found in any given postsynaptic density at different levels. And so one of the complex issues with looking at neurons is that each postsynaptic density is completely unique. And Seth Grant did some really nice calculations and found that there are at least 14 trillion combinations of molecules that can occur within any given postsynaptic density. And so, understanding each postsynaptic density will be very difficult. However, we can look at general principles using some of these proteins. And so Ming-Jae Zhang actually did some very nice work earlier in the late 2010s, showing that protein fragments of each of these scaffold members can lead to the condensation on the membrane, linked with an NMDA receptor subunit tail. He showed that through their reconstitutions that model PSDs can interact with and promote polymerization of actin filaments, just as I showed just as Chalet and I showed in T cell at the T cell synapse. And this is dependent on chordactin and the ARP23 complex. He also showed that model PSDs can sort molecules, much like we saw with immunological synapse and lac clusters in CD45. In the model PSDs, CaMKII 2, which is highly enriched, maybe the most abundant protein in the postsynaptic density, is enriched in these condensates, while Geoffrin, which forms inhibitory condensates, is excluded. And in a follow-up paper from his group, Ming-Jae and his colleagues showed that this is driven through electrostatic repulsion, just as we saw with latent CD45, really, reinforcing that we can learn a lot about neurological condensates from general principles established in immunological condensates or immunological synapses. Okay, so my group, when we started in 2019, right before the pandemic, which was excellent timing, we decided one of the goals was to reconstitute the postsynaptic density, but not using protein fragments, as I'll get into a little bit, but using the full-length proteins. And so we set out to purify some of these proteins. Homers and McGucks have been, purification has been established. However, DL-gap or GCAP and Shank proteins has, to the best of our knowledge, never been done before. So, starting in the pandemic, let's purify proteins that have never been purified before. That's great. So long story short, we've been able to purify DL-gap, shank2, and shank3. And what we found is that DL-gap on its own can undergo phase separation. It quickly forms a gel. And as you'll see, shanks2, and then you won't see shank3, we can purify the full-length version. And we can work with it. We can phosphorylate it. We can do different modifications on it. However, they also form condensates readily. And I'll get into that in a bit. And so that led us to really rethink how we're going to study this, because if each of these proteins can form condensates on their own, we get into some complex coacervation rapidly. And what we really wanted to do was link some shank functions with downstream, some shank mutations and functions with downstream effects. And so what we did is we minimized the system using only Homer and Shank. And so these, even though the PSD is a membrane-associated condensate, we're looking solely at in-solution information here. And so the first thing that we did is we asked whether Shank2, much like Shank3, as Mingji showed earlier, could undergo phase separation. We see it at 50 nanomolar. And so using the full-length protein, these shank2 and Homer1 undergo phase separation at very, very low concentrations, but they are physiologically relevant. And so we then asked, well, these are spheres, they must be dynamic, right? And so we performed FRAP, and as you can see in the recovery chart, there's no recovery. So we photo bleached a section of these condensates, and then over the course of 10 minutes and beyond, there's little rearrangement, if any, and there's no exchange of molecules from what we can see with the outside environment. And so what this at least suggests is that these condensates likely form as a liquid, minimizing the surface to volume ratio in spheres, but then they rapidly, within about a minute, transition to a gel-like substance. And so phosphorylation is a common modification. We know that based on online databases that shank2 is able to be phosphorylated at many sites. And so we chose MAP kinase to phosphorylate shank2, and we hypothesized that maybe phosphorylation will regulate the dynamics of these condensates, as well as possibly the formation. And so we performed experiments with phosphorylated shank2 and Homer1, and what we found is that phosphorylation does very little to alter the dynamics and formation of these condensates compared to the non-phosphorylated version. And so to me, this was kind of disappointing because, in my training, in my signaling world, phosphorylation does everything. And here it appeared to do absolutely nothing. So that was kind of a bummer. But what we have is a usable platform that we can now perform functional experiments with either phosphorylated protein or non-phosphorylated protein. And so the shank family proteins have historically, canonically been linked with actin polymerization and controlling spine morphology through increase or decreasing actin polymerization, depending on the rate of signals coming in from the presynaptic bouton. As I mentioned, we had a bit of downtime because of COVID. And so we did some sequence analysis with Dr. Foreman K here at Sick Kids. And what we found is that shanks 1 and 3 gene ontology terms linked with their intrinsically disordered regions were clustered very well with actin machinery, suggesting a direct link between the gene ontology of their IDR and downstream function. However, shank2 surprisingly contained Go terms that were linked with RNA translation. And so search the literature a little bit. There's been some work on shank2, not nearly as much as with shank3, but some of those studies found that there were translation differences in neurons that express shank2 mutants. And so we thought that maybe RNA translation in the spine is linked with shank2. And so shank2 knockout neurons are hyperconnected, suggesting that there's additional RNA translation to generate these proteins that occur when shank2 is not there. So maybe shank2 has a role with dampening or inhibiting RNA translation. Shank2 mutations also alter the global translation, continuing with our idea that maybe there's a link between shank2 and translation. And importantly, the computational analysis showed that shank2 actually clustered with FMRP and CaPRIN1, which the Foreman K lab just a few years ago showed to regulate RNA metabolism. And so we thought, hey, this is great. Here's a link. Maybe shank2 can regulate local translation through FMRP and CaPRIN. And so why might that be? And so just a brief simplified look at FMRP regulation of translation. So Julie's lab did some really nice work looking at how different forms or different modifications affect FMRP-dependent translation. What they found is that when the IDR of FMRP is phosphorylated, translation is dampened compared to non-phosphorylated version. And then when it's methylated, translation is increased. And so simplified version is phospho-FMRP suppresses translation, FMRP promotes translation. Importantly, the kinases and phosphatases that act on FMRP, specifically PPA2 and CK2 are localized specifically within the postsynaptic density. And I just want you to think, for the purposes of this talk, that FMRP is a switch. So upon phosphorylation, it suppresses translation, and then upon phosphatase activity, it can return to its non-phosphorylated state and then promote translation. So we asked, does shank2 with molecular features similar to FMRP modulate RNA metabolism, in this case translation, through interactions with FMRP? And so once again, we work with our shank2 and Homer model simplistic system, and we asked 3 questions. So number 1, does shank2 regulate translation? Number 2, if yes, how? And then 3, do disease-linked mutations alter shank2 modulated translation, as has been suggested by some of the observations in neurons that express shank2 mutants? So our hypothesis is that it's the phase separation specifically of shank2 that regulates RNA-binding protein localization, FMRP, and the phosphorylation state of the RNA-binding proteins to control translation. Furthermore, disease-linked mutations in shank2 can alter these dynamics. Okay, so our first question was, do shank2 condensates affect translation? So what we did is we used a luciferase translation assay. We pre-mixed shank2 condensates, dropped them into the rabbit reticulocyte lysate, allowed translation to occur, and then measured the amount of luminescence from luciferase following translation. And what we see is that in our control sample, which contains only luciferase RNA and rabbit lysate, we have a lot of luciferase translation as indicated by its luminescence. However, as soon as we add shank2 and Homer1 condensates to that mixture, the translation is dampened by about 5 orders of magnitude, showing that shank2 does something to inhibit translation as we have hypothesized. And we did some microscopy, and we did show that these condensates remain even though they're moved from just a solution-based to a lysate. So they're very robust condensates. Okay, so they can modulate translation in vitro. And then we asked, do disease mutations alter the enrichment of FMRP or phospho-FMRP? And so the first question is, does FMRP go into condensates? So we used a physiological buffer and looked at whether FMRP could localize to shank2 condensates. And at 150 millimolar salt, we saw very little enrichment. There might be a very little bit if you look closely, but by and large, FMRP remains, it doesn't really detect the condensates. However, at 50 millimolar sodium chloride, we see good enrichment of FMRP in the condensates, suggesting that electrostatic interactions drive its enrichment in condensates when there's low salt shielding of whatever interacting motifs there are between FMRP and shank2. So then we asked, because it's electrostatic, maybe increasing the negative charge of the intrinsically disordered region of shank2 will affect FMRP enrichment. And so we did several experiments in which we looked at phospho-shank condensates, phospho-FMRP, and then different combinations of the two. And as you can see from our measurements on the right, there's no significant change of FMRP enrichment in shank2 condensates, regardless of phosphorylation status of either protein. And so once again, my whole thought process that phosphorylation matters for everything was completely blown up, which is great. I don't mind being proven wrong, but it also makes us wonder, well, how, if phosphorylation is not really doing anything in these wild-type condensates, does it matter for function? And so that's just a question that I want you to remember as we go through the rest of this talk. So we've showed that FMRP can be enriched in condensates. What about RNA? So RNA has never really been detected in the postsynaptic density. There's been some omics work that's found that microRNAs can localize there, but by and large, the postsynaptic density is thought to be a protein-rich environment. However, if you look at electron micrographs, you're not going to be able to see RNA anyway. So whether it's there or not is still is not clear from cellular data. However, in vitro, we can see that RNA readily enters shank2 condensates when FMRP is present. And so all of this data allows us to build a model in which we think that shank2 condensates are regulating RNA translation through the absorption of FMRP and mRNA preventing translation from occurring outside of the condensate. And so then we asked, okay, this is great. We have an experimental platform. We can look at different aspects of it microscopically. We can look at functional outputs through translation assays. What about disease-linked mutations? Because there are a lot of them. And so each of the lines here show different mutations that have been sequenced from patients in shank2. And shank2 is now recognized by the Simons Foundation as being autism. It's highly correlated with autism. And so today I'm going to focus on 3 specific mutations. First, I'm going to talk about the R958S and A1731S. And then we have a patient line of induced pluripotent stem cells from a patient that has a heterozygous truncation mutation. And so we're asking whether we can apply what we've learned biochemically to an actual patient's cells. And also importantly, a lot of these missense mutations are found in the intrinsically disordered region. And so shank2, the isoform that we're using is about 1,450 residues. And so the obvious question is, what will a single point mutation do amongst an IDR of about 1,000 residues? And Rick Young just released a paper looking at missense mutations in different condensate forming proteins and found that just a single missense mutation can alter its localization. So just keep that in mind. And so we first looked at whether FMRP localization was altered by these mutants. And so on the left here, if we look at dephosphorylated proteins, we see that FMRP is well enriched in the mutant condensates. Phospho-FMRP enrichment doesn't really seem to change very much when normalized to shank2 intensity. However, when the 958S mutant is phosphorylated, we see a drastic reduction in FMRP enrichment in the condensate. And also, this will come up later, but note that it seems to only enrich the edges of the condensate. We also looked at the 1731S mutation, and we saw the exact same phenotype, in which when shank2 mutant was phosphorylated, FMRP is not very well enriched, and any enrichment seems to occur at the edges of the condensate. And so if we look at, just to compare visually, we see good enrichment with wild type, good enrichment of FMRP and wild type condensates. However, in these mutant condensates, when they're phosphorylated, there's little enrichment. And this plays out very well when we quantify the relative fluorescence of FMRP compared to shank2 in the condensates. And so there's a significant decrease in FMRP. And so we also wanted to look at RNA and whether the pattern of RNA enrichment follows FMRP enrichment. And what we see is that, yes, when when the mutant condensates are phosphorylated, the amount of RNA within the condensate also is diminished compared to when normalized to shank2, just as FMRP. Okay, so here comes a so what, who cares? This is great biochemically. We're using, we can use mutants and look at localization of different proteins, but what about in neurons? And so what we did is we teamed up with James Ellis group here at SickKids and looked at induced pluripotent stem cell derived neurons at 4 weeks. And so the key with these experiments is that we're looking at a neuronal environment, not so much that we're looking at dendritic spines. We're doing that work now. And so what we found is that when we express mCherry shank2 and mneon green FMRP in either shank2 wild type or shank2 knockout neurons, we get good co-localization of the 2 proteins together, which is very similar to what we see in vitro. However, when we express the 958S mutant, we can see a complete localization change of FMRP. And importantly, it doesn't matter if there's wild type copies of shank2 in the background or not, the 2 proteins don't co-localize with each other in these neurons. We see the exact same molecular phenotype with the A1731 mutant, in which FMRP does not co-localize well with shank2. And when we just ran Pearson correlation, we can see a significant difference in the co-localization of FMRP with shank2 condensates in these neurons. Importantly, this also suggests that in a heterozygous situation, as you'd have with a wild type neuron, that the mutation dominates. And so even though, which is important because in the disease, these missense mutations are heterozygous in the patients. And so these data suggest that even though you have only a heterozygous missense mutation, it can dominate the behavior of condensates in cells. And as I mentioned, we're currently working on we're currently performing functional assays looking at the functional result of this mislocalization of FMRP in these neurons. Okay, so going back to the biochemistry, as I mentioned, it seems that with mutant condensates, FMRP localizes only to the edges of the condensate. This at least suggests that there might be a physical change in the variant containing condensate. And so the question comes up, well, how do we study this? So one of the obvious experiments to do is looking at fluorescence recovery. But as I showed you earlier, within about a minute, these condensates turn into a gel. And so we have a technical limitation in that we can't really look at the dynamics of molecules in these condensates simply through FRAP. But here at CaMKII, we have an oscillation rheometer, which allows us to look at the viscosity and elasticity of these condensates. And so looking at the viscosity of the condensates using the rheometer, what we find is that the disease-linked mutants are more viscous than the wild-type condensate, even though they're in a gel-like state. And when we look at elasticity, we see the storage modulus, which is an indication of the ability of the material to store elastic energy. We see that both mutants have a higher storage modulus than the wild-type condensate. And if we look at the loss modulus, which is the amount of energy dissipated, we see that there's a distinct difference between mutant condensates and wild-type condensates. So this allows us to conclude that even though the dynamics within the condensate are very, very low, that mutant condensates are more viscous and elastic than wild-type condensates. And so we're able to build out this model now that includes the disease mutations, in which we see that phospho-FMRP is well enriched in mutant condensates. However, RNA and FMRP are diminished, and that's linked, at least correlated, with an increase in viscosity and elasticity compared to wild type. And so the functional output of these condensates, if RNA translation occurs outside of them, is that RNA translation should go up because FMRP, non-phosphorylated FMRP promotes translation, and its phosphorylated counterpart that inhibits translation is mostly in the condensate. And that's exactly what we find. And so when we reconstitute mutant condensates in our rabbit reticulocyte lysate assay, we find that translation is increased 15 to 30-fold depending on the mutant compared to wild-type FMRP, or wild-type shank2. And we think that this is the first molecular insights into why shank2 mutant neurons see an increase in translation. It seems that some of these missense mutations, at least this class of mutation in which serines are generated in the intrinsically disordered region, can lose their ability to inhibit translation, which is its wild-type function. Okay, so I mentioned that we have a patient here at CaMKII that is heterozygous for an R841X truncation mutation. And we thought, well, we have the iPSC, let's take a look at FMRP localization. And so what we see is a pattern very, very similar to the missense mutations that I showed earlier, in which here we express wild-type shank2 in these neurons that have that are at least genomically have a truncation mutant, and what we see is a complete mislocalization of FMRP away from the condensates, just as we saw previously. So then we asked the question, what about FMRP and phospho-FMRP levels? So if you recall in our model, depending on the ratio of the two, you'll either promote translation or inhibit translation. So if you have more phospho-FMRP outside of the condensates, you'll inhibit translation. And so in these neurons, when we run Western blots, we see that the ratio of phospho-FMRP to FMRP is actually increased, meaning that there's more phospho-FMRP in these neurons. And when we rescued the R841X mutation by replacing it with a complete copy of the shank2, we can see the ratio return to wild type levels. This allows us to build this model in which we have high FMRP and phospho-FMRP outside of the condensate, thinking about our localization data. And so what we should see is a diminishing of mRNA translation experimentally. And that's exactly what we see. So we use CaMKII, which is an FMRP targeted mRNA as the indicator. And what we see is that in the R841X mutant, which has more phospho-FMRP available, CaMKII translation is diminished, matching our model. Importantly, though, the mRNA counts are very similar between the two cell types. So your rescued cell type has the same number of RNA molecules as the mutant cell type, indicating that this regulation occurs at the protein level, providing more support for our model that shank2 can regulate translation and that disease-linked mutations in shank2 can alter that translation downstream. Okay, so just in summary, we showed that full-length shank2 and Homer 1 undergo phase separation at physiological concentrations, and these condensates are not dynamic. They do not recover well following photobleaching. FMRP can be enriched in shank2 condensates, and that enrichment is sensitive to cell concentration, suggesting that electrostatic interactions are the driving force behind its enrichment. These condensates can repress translation in vitro. Mutate the 958S and 1731S mutations dysregulate mRNA enrichment both in vitro and in neurons. These mutations also lead to an increased viscosity and elasticity of the condensate compared to wild-type shank2. Expression of these mutants in wild-type background cells shows similar aberrant FMRP enrichment, suggesting that these mutations are in fact dominant, which really provides a mechanism for understanding missense heterozygous mutations that are often found in patients. Phosphorylation of the shank2 mutants promotes RNA translation compared to wild type, which also provides some insight into the mechanisms that explain observations in neurons. But also the truncation of shank2 that deletes the IDR alters the ability of it to interact with FMRP, and it leads to a dysregulation of the ratio of phospho-FMRP to FMRP, leading to repression of CaMKII translation. And so with that, I'm happy to take any questions you might have.
Thank you so much, John. We have a question from the chat. Mikhail, would you like to unmute and ask your question?
Thank you for the very interesting talk. I'm just curious from the general point of view, what is the protein concentration inside these droplets when you do them in vitro or when they are inside living cells? And basically, are they more dense than the cytosol or less dense?
Right. So I'll give you a two-part answer. The first is that we haven't measured the concentration within the condensates of shank2, and that is because there's a technical limitation of the microscopy we're doing, in that the background fluorescence when we image the condensates is low enough that it's within the noise, and so we can't actually calculate the ratio of enrichment for shank2 in the condensates. We could do some fluorescence calculations and actually get to that as we've done with other condensate systems. So, and then the second part is that in the neurons, there's not a uniform concentration of shank2 across the cell. So because these cells are so highly polarized, the proteins are not well expressed in the soma or body of the neuron and then transported out or diffused out to the dendritic spines. The dendritic spines are actually the centers of protein translation. And so within any given dendritic spine, you're going to have a higher concentration of shank2 compared to anywhere else in the neuron. In terms of the physiological concentration, I'm not sure what it is for shank2, but I can tell you, so if you look at another postsynaptic density protein, Ming-J Zhang has said that PSD95, which is highly enriched in the postsynaptic density and dendritic spines, is roughly 100 micromolar. And so, even if shank2 is 100-fold less than that. It's still at about a micromolar in the dendritic spines, which is well above the critical concentration for phase separation on its own.
Thank you. Any questions in the kitchen table? Oh, Cameron has one.
I have a question. Excellent talk, John. Fantastic work. I was wondering if, so, you know, you had mentioned that shank1 and I think shank3 was the one that you couldn't purify. Did you guys have a look at these other two shanks? And in addition, is there any known mutations that are linked to these diseases in these proteins?
Sure. Okay. So first question. So we've attempted to express shank1 and shank3 in bacteria. Shank1, we have been unsuccessful. It only expresses like the first half of it. And then we don't actually know if there's like some rare codon stop or something. So we'll probably do that in insect cells. Shank3, there's actually a student in my group, Gaddy, who has successfully purified shank3 using similar methods that we use for purifying shank2. And he's found that truncation mutants have a very different phase behavior than the full length. And he's actually done some bio-ID in cells with Jiyoung Yoon, showing that missense mutations in shank3 alter protein networks, specifically looking at actin machinery. And so different missense mutations in shank3 alter probably the condensate composition with regard to actin, which actually fits with those mutations because there's changes in actin polymerization and dendritic spine size in neurons that express shank3 mutants. In terms of diseases, these two mutations, the 958S and 1731S, have been found in autistic patients as well as schizophrenic patients. And so depending on various genetic factors and also protein expression factors, the presentation of disease can be different even though the same protein is mutated.
Got it. Thank you. Yep.
We have a question from, oh, sorry. There's one question online and then you. So Lindsay, go ahead and ask your question.
Hi, John.
Hey,
Really
Lindsay.
nice talk. I was just curious, like, it's really interesting that the mutants are often have a phenotype in the heterozygous background. And have you explored that at all biochemically? Like if you mix your wild type shank with the mutant shank, do they co-condense? Does the mutant like dominate the viscosity and elasticity, or is there some like intermediate material property? And how have you explored that?
Yes, so we haven't explored it. It is on our list. And so that is actually on our list to do because, you know, in cells, we see this dominant effect. It'd be interesting to see if a similar effect happens in vitro, for sure. These are great ideas.
Anthony has a question here.
Hi, John. Great talk. This question could be a little far-fetched, but just that you mentioned the connection between shank2-driven condensates with autism and some schizophrenia. So you're in the hospital, you have samples. Could this work be extended to finding or detecting in biological samples like blood, CSF, and used as a kind of a biomarker to track or diagnose this kind of things where you find this connection and this very interesting connection?
Yeah, absolutely, absolutely. From this, I think along these lines, from these experiments, we're actually seeing that there may be classes of mutations that have very similar, physiological patterns, even though the mutations are very distinct. One of the intractable things about studying the postsynaptic density and thinking about it in terms of correcting or rescuing the disease is that there's tens of thousands of individual mutations in the 600 or so proteins that have been linked with disease. But if there's individual classes of mutations, and maybe we would get more information by looking at the variety of patient samples that we have access to, we might define classes that could be potentially corrected using a single corrector, whether that's a peptide or a small molecule. It would be actually interesting to be able to classify postsynaptic density linked diseases in different bins rather than looking at each mutation individually.
Thank you.
I have a question. That was a wonderful talk, John. Thank you so much. So I was curious, these shank2 Homer FMRP condensates, is the effect on the translation global, or there's some form of selectivity?
Right. So in the in vitro assays, it's global, but I think in the neurons there is some structural selectivity built into where these proteins are actually expressed and where they condense. And so FMRP is packaged into RNA transport granules and then shipped as part of the translationally repressed RNA to the polarized regions of the cell, whether it be the axon terminal or dendritic spines. There, the RNAs that are transported are translated locally. And so so if you think about a whale neuron, that can be up to 30 meters long. You're never going to be able to get a protein from the soma out 30 meters. And so to overcome that, they ship the RNA and then translate it locally. And so the experiments previously that have looked at expression of proteins have looked at it globally, but in reality, it's really only happening in the dendritic spines by and large. And that's simply through the localization of where these condensates are, and then diffusion barriers driven by the structure of the neuron.
Great, thank you. Any other questions? If not, I have one more. No? Okay. So going back to the story of filtration and exclusion of specific compounds or proteins. So in the first example, you talked about electrostatic repulsion. I was wondering, is that sort of the general rule, or how does the lattice size contribute to that? Has anyone looked at it?
That's a very, very good observation. So electrostatics, at least from what people have looked at, and maybe they've looked at it because it's the easiest thing to look at, is a very large contributor to the sorting of molecules when there's not specific binding motif interactions or domain motif interactions. We originally thought with CD45 that maybe, CD45, which is has a fairly large intracellular domain, is unable to penetrate because the lattice size is too small. The effective pore size is too small to allow it to be in. We didn't rule that out as a possibility because the electrostatic effect was so large. When we used just generic proteins with different charges on them, we were able to move these generic proteins either outside or inside, depending on their charge. So it could very well be that the intracellular domain exclusion is linked to both charge and size. We don't know. Yep. Some of the really nice work with microreology on microscopes shows that different dextrans with different sizes can either penetrate or not penetrate depending on the effective pore size. And so that's absolutely a possibility with sorting molecules as well. I will say with postsynaptic density, so CaMKII is a gigantic hollow enzyme, 12 subunits. That's readily enriched in the condensates, while Geoffrin, which is much smaller, is readily excluded. So that's probably the postsynaptic density, even though it gelates very rapidly, I'm not sure the effect of the pore size there. But I think it is a very viable potential regulator of sorting of molecules.
Okay, thank you. Very interesting.
I have one question, actually. John, I have a question. So one thing I noticed throughout the talk, right, is that the proteins, as you mentioned, don't change at all when you change the phosphorylated state.
Yeah,
but then with these mutations to serine threonine, you have this whole story, right? Yep. So question for you, moving forward, whenever you see a mutation that drives disease, and it's to a serine threonine, very intriguing that it's going to, have an effect on phase separation and condensate behavior. So,
yeah, and so there's actually several serine threonine mutations. We just looked at these two, but there are several within the shank2 IDR. One of the difficulties with working with shank2 is that it has not been studied very well. And so the reason that we used MAP kinase to phosphorylate the protein. One of the issues is that we did that based on predicted phosphorylation sites. So MAP kinase experimentally, as far as I know, has not been shown to phosphorylate shank2 at all. So, and it only phosphorylates very specific residues. And so some of the serine 3 mutations don't lie within, say, a MAP kinase predicted phosphorylation regime. So it's a technical limitation because, we're an academic lab. We can't test all 30 kinases that are localized to the PSD to phosphorylate some of these sites. But based on what we've seen in our experience with some of these mutations is that that mutation likely will change the behavior of the condensate, whether through compositional changes or, or physical changes. And then it just, because there's so many kinases there, there's a high likelihood that it would be phosphorylated.
Right. Cool. Thank you.
Yep.
Thank you so much, John. If there are any other questions, feel free to submit them through the contact form on condensates.com. Join me in thanking John again for such a wonderful talk.
Awesome.
And the recording is going to be available on condensates.com within the week. Have a great day, everybody, and see you next time.
Thank you.
Bye.